Michael Jackson.
Thank you. All right. So I always find it amazing that drugs work at all. I find it quite incredible, the fact that you can design a small molecule and have people eat it, and it's going to do something good rather than bad. Look at the complexity of the human body. It's enormous, the number of different proteins that are present. There's proteins, there's carbohydrates, there's lipids. You introduce a small molecule, and it's going to hone in and bind one particular or a few proteins in amongst all the ones it could bind, and do something useful rather than something toxic. We struggle constantly trying to come up with new drugs, and it's very much because of that. It really is an unusual thing to find. It's unusual to find amongst people calculate the 10 to the 63 different chemical compounds that you can make by organic chemistry. But amongst that huge chemical space, you can actually find those that will do something useful as opposed to something toxic. And to make matters worse, all the testing that we can do in animals tends to be rats, dogs. They're not humans. They're different. Not only that, humans aren't inbred enough. You're different. Just to make things a little bit worse. So, no sooner have we done some trials with compounds and it seems to be safe, there's always the next person who is different, who then will have an adverse reaction, and sufficiently severe that the drug is not going to be marketed. So, not wanting to be depressed, we've sort of got a lot of new technology to help us these days, but you have to also recognize that the track record is not looking too good at the moment in terms of pharmaceutical companies spending more and more money on doing more and more research and coming up with fewer and fewer drugs. Not a pretty picture. I mean, particularly if you're in the pharmaceutical industry, right? But even for you, and for me, and for everybody who's out there with serious diseases, it's not like we've cured many diseases. There's a whole load of them out there. We've heard a lot about them today already. That's a lot of need out there. So what are we going to do about it? Well, I'm sure you, like me, are involved in the technologies and hoped and dreamed that when the human genome came out, we'd, that might make quite a big difference. And in fact, we got sneak previews of the human genome from sequencing of ESTs and all the rest of it. And I remember trying to convince my management, and I know that many other companies and biotech, we all went along and said, "Oh, well, just give us this, and we'll come up with, we'll find new targets and we'll find new drugs, and it's not going to be that difficult, is it? Yeah, well, things moved on, and I want to tell you about a few of the technologies today that we and others have been using that I think are really amazing in terms of what has happened since the mid-90s to where we are today in terms of the industrialization of the biological sciences. The huge amounts of data we're making and generating. Let me start off with the first slide here. Oh, it's already there. Okay. It's here. We can argue about the numbers of exactly, apart from the organisms, there's only one organism that's in this slide, okay? You can argue about some of the other numbers, and we're scientists, we do, all right? How many cell types there are, how many exactly genes there are and proteins there are in the human body. But this gives you some sort of numbers, right, to sort of scale to. And the human body is about 70 trillion cells. Actually, looking around, I think some is a few more than 70, but, by and large, that's the right sort of number, right? I don't know where he went to, but... where are we going to tackle this system? We can tackle it down at the organism, the old way we used to try and find drugs. One of the, feeding compounds to rats and to people. In the systems areas down here, not so complicated, but you couldn't really get traction on it. Going up through here, where is the level we should play at? Where is the level we should be looking to collect data at these days? Well, a lot of people now are focused in at the top level up there. We've got the human genome. What are we doing with it? How are we using that information, leveraging the information from the human genome to help us find new pharmaceutical products? Well, one of the things we focus on particularly is at the cellular level. You heard from one of the previous speakers about microarrays. These are amazing devices, tools, little microscope slide with 10, 20, 30, 40,000 different genes arrayed down there in a particular pattern. And what you're able to do is take a sample and basically ask in that sample which of the genes are expressed. So if you have some brain, for example, you could mash up the whole brain, extract it, put it on this little array, and it would tell you which of the genes from the human genome essentially are being expressed in the brain. Well, you can do a geography map of what's where. For example, if click on here. Here's a section of some nerves. In fact, this is the dorsal root ganglia. I think someone was talking about a dorsal. This sort of junction box from the periphery of your nerve going in towards the spinal cord there, a junction box. You've got large cells, small cells, whatever. Our very first experiments of geography that said, of all these new proteins that exist, where are they expressed in the human body? We just took things like brain, liver, lung, heart, and did a real crude geography experiment and said, where are all these things, and build a database, okay? And so now I've got a new possible target of the drug. It's only expressed in a particular, so say it's only expressed in the heart. Maybe that would be a useful thing to go and look for in terms of targeting with small molecules, small drugs. Very crude filtering to start with. We pretty quickly learned, though, that that's not what people want. They don't want to know exactly which cells all these new proteins are expressed in. Because only with some level of precision, down to exactly which cells, can we start to ask more precise questions. So we hooked up with, and other people have done this, you hooked up with a company that sell these it's invented at the NIH, a machine that actually allows you to laser capture. And this, now we're down to individual cells, but if you look at the large cells shown here in the green, you can actually use a laser to essentially fire the laser at the cells you want. They stick it onto the microscope slide, and then you can, well, stick it actually, it fires it onto the lid. We'll see it in a minute. And you peel off like hairs off a Band-Aid. You can actually, you capture the cells, right? And then from the same section, of course, you could capture the small cells. Now, that's all very well, but what you really want to know now is what are the proteins that are specifically expressed in just those things. If you think about the brain, it's particularly important, right? In the brain, you don't want to mash the whole brain up and say what's expressed in the brain. You've got all these nuclei and all these particular connections. You want to know exactly which cells in the brain these particular proteins are expressed in, so I can start building hypotheses about what they may do. And using this technology in track tracing, you can get now down to actually following circuitry, inject at one part in the brain, let the dye travel down to a nuclei, and then you can actually, using the laser capture those individual cells. Now the amazing bit to me is with a single individual cell, you can get enough RNA, in other words, the component of the genome that's transcribed that actually is going to make the protein. You can get enough RNA from one cell to then go ahead and put it onto a DNA microarray. Now we've got single-cell expression data. I put a little movie together here, which probably, we can click on it. Just shows you the whole overall process here, which we actually, this is being highly roboticized, collecting basically, going into tissue bank, getting your sample. There's all sorts of applications, taking it off, chopping it up in the microtome. In other words, cutting very thin sections of this stained section. You look on the microscope, look at the cells, see how they look, and then decide that it's a reasonable section. Then determine the cells you actually want to capture with the microscope, the laser microscope. Fire the laser actually at the cells. A couple of companies doing this, Arcturus, Palm, actually now have this. And you can see, fires the laser beam down onto the cells, one cell thick, just capture an individual cell, in this case onto the lid of an Eppendorf tube. A single individual cell, then you put extraction buffer, extract the RNA from that. Corny, but anyway, but extract the RNA, okay, it looks like DNA. And then you actually label it up and stick it onto a DNA microarray, put it all into the database, and those are supposed to indicate the microarray, of course, the more the gene is expressed, the more the color. And then we extract all that, put it in a big database, and try and sort through all the data. Now, our problem is, we've got huge amounts of data. Just huge amounts. I mean, the labs are, you go around the world, you're finding people are collecting 1 million, 2 million data points a day of this type of what's expressed when and where. Well, Excel runs out after 60,000 lines, and it gets pretty aggravating, right? I mean, not only that, I have no idea what most of the proteins are. We have not trained our scientists to deal with this stuff. We don't know how to do it. we have to turn to other people to help us deal with this type of information, how to visualize it. Just shown on the next slide here, this shows essentially 20 million data points in one shot. Now how it kind of works is, across the top you've got 2,000 different cell types, which we've analyzed by this process, and down the side, 10,000 different genes. And if a gene is expressed at high levels, we're indicating it in red. If it's at low levels, it's in blue. And in this case, we move the cell type next to each other that has the most similar expression pattern by target, and then you put the targets next to each other that have the most similar expression pattern by cell. So you do double cluster analysis, and by this, you can now use this sort of thing for some pretty amazing things. You can come along to anybody now with a single cell, and I'll tell you where it's from in the body exactly. No problem. I'll tell you with absolute precision where it's from in the body. I'll map it into this matrix. Look at the power of this type of thing now. Obviously, many people in the world are now using this type of approach for cancer diagnostics. You can imagine how you can go in very early on, the most important time to try and diagnose cancer, go in, get a small number of cells, or at the very early stage, analyze them, see whether they have a normal, are they going to be benign, or are they going to be tumorigenic? What's going to happen? And we start to make predictions based on historical data. This is pretty exciting, but not that easy to actually find drug targets out of it. That's been our challenge and many people's challenge. And just dealing with this type of information has altogether challenged us, because a lot of these genes, there's a huge amount of literature about it, and we don't know how to access all that information one at a time. We have been trained as reductionists. We've been trained in our PhDs to go and work on a single molecule and become a world expert on it. That's what's been going on. Right? We have much fewer people have been trained to be working on systems. And it's quite humbling, right? You realize how terminally differentiated you are as a human or as a scientist. You know, you've just gone down this road. So we started to turn to other helping technologies to try and mix together this type of microarray experiment, or this gene expression information, with all the information that's in the literature. Let me give you an idea of how this works. This is actually an experiment that involved human beings. What we did was we administered LPS, which is a bacterial lipopolysaccharide that gives a mild fever to them. We then extract blood out of the patient at various different time points shown across the top, 0, 2, 3, 10, 24, from 3 different donors here. Then we put the blood on the microarray and say what's changing. Again, we can then cluster analyze this by resorting out into various different, those genes that behave the same way, either over the time period, either going up in expression or down in expression, you cluster together. And you say, is there anything I can use from that information? Is that knowledge of those collections of genes that are expressed together has ever been found before? Well, each of these different genes already has some key features about them. In fact, if I take one of these clusters here, let's just take one of the clusters, cluster 8 here, and start looking at the genes in there, and you will find here, and this is a cluster analysis relative to keywords, so of the genes in this cluster 8, you'll find that many of them had an indicator that they may be involved in inflammatory response. Well, so what, you know. What you can do now is go to the literature and go to Medline and capture all the data to do with inflammatory response, all the Medline abstracts that have to do with inflammatory response. Okay? And then you normalize them all and retrieve them all, and then you put those on what's called a galaxy view. So this is a view of all the Medline abstracts to do with inflammation, shown by similarity to one another based on either keyword or a moving window. So just looking for rare characters in those abstracts. So things that cluster together are abstracts of papers or papers that have similar content. Then what we've done is taken the genes in that cluster number 8 and mapped them so do any of those occur in any of those abstracts. And what we find is, sure enough, in some cases, you find that you have a set of abstracts or papers that have 2 or 3 or 4 of those genes in there. This is a way that we are able to basically turn huge amounts of data, use pre-existing information that's out there, mix it together and say, when I see those 5 genes, that was already known in the literature. I can actually, therefore, extract that. Otherwise, you spend hours and hours and hours on Medline It is just not tenable. We have got to find ways of extracting. We have got to find better ways of using this data. We are collecting far too much data and getting too little information out of it. That is really our greatest challenge, to be able to narrow down, to use this effectively. I want to sort of end on another area entirely, which is around the use of modeling, computer modeling of physiological systems. For those of you who actually studied biochemistry at any level, you probably remember those horrible charts on the walls where everything was connected to everything else and you could never remember most of it, right? They were just huge. Big circles all over the place. I think I learned the TCA cycle, but that was about it, right? I still think I couldn't do it today. If you actually go, there are mathematical models. I remember the ATP was always involved everywhere, right? That's what I do remember, and you could never really figure it out, right? But you can imagine you can model systems. You can either model systems in biochemical systems, you can model systems involved in physiology. Our greatest challenge today in finding, in moving drugs to the clinic is actually that despite all the experiments we do in animal models, all the experiments we do to test toxicology, it's just guesswork at the end almost. I mean, 50% of them just go off, and they go either way. You find compounds that as soon as you administer them to humans, they're 50 times more active than anything we've ever seen before. And the other direction. More, unfortunately, more frequently, but that's the way it works. So we are not able today to predict what's going to happen. We're also not able to predict with great certainty at all what's going to happen with which patients are going to benefit the most from our drugs. So we've teamed up, and there are other companies, with a company called Antelos, and also with the American Diabetes Association, and other companies are working on this also, Physiome Sciences, to try and build a physiological, mathematical model of diabetes, involving all the organ systems, involving all our knowledge base that we have about down to the individual proteins and how they connect, and when I do this to this, this will happen, when I do that to that, and you actually have black boxes, and you can build virtual patients, right? So you can have people who have a certain weight, certain age, certain lifestyle, certain this, and you can go play with it. Our dream, of course, is that we will be able to essentially predict that when a human takes a compound, we will be able to predict what's going to happen in their body when they take that pill, what's going to happen in all the organ systems, what's going to happen in the liver. We're going to be able to measure and we're going to monitor what's going to happen in the kidneys, what's going to happen in the brain. We're going to be able to predict what's going to happen in terms of concentration of drug, what's going to be the outcome, and we're getting there. This is a hugely complex mathematical problem. There are supercomputers that actually are running the mathematical equations behind this. Right now, they don't work that well, but they do work to a certain extent. They work at a crude level. They are helping people being able to design clinical trials. They are helping us take the information we get from our clinical trials for our diabetes medicine and try and plug that data in and say, does it make sense? Am I treating the right population? Am I treating them at the right time? And, this is really a whole new area, the idea of having virtual patients. And it goes a step further. It's as good as the data you put into it. We are starting to now look at ways of getting much better data streams about people's glucose levels, about other measurements, real-time measurements. You can imagine having implanted wireless devices that will give you real-time glucose measurements along with other measurements of what's going on in the human body. They will be sent quite simply to a central database at which you then will have the model which will then tell you what you should be doing next, how much insulin you should be injecting, what you should be eating, what this, what that. I don't think that's that far away. I don't think that's that unreasonable to assume that could happen. Will that give us better management of diabetes? Maybe. But it certainly will be using the full power of the data we have. And to me, that's our greatest challenge today. Thank you very much.